Mutations. Changes in genetic material or the genome of an organism is called a mutation. They may occur spontaneously or due to external factors such as UV rays, heavy metals, or chemicals. Mutations are transmitted from one generation to another. Two main types of mutations are gene mutations and chromosomal mutations. Gene mutations. Gene mutations occur due to errors of DNA replication. Order of nucleotides or the sequence of bases are changed in gene mutations. These gene mutations can occur in several ways.
They are substitution, insertion, and deletion.
Substitution. One base or a nucleotide of the DNA sequence is replaced by another base or a nucleotide in substitution. Different types of mutations can occur due to substitution.
They are silent mutation, missense mutation, and nonsense mutation. We can clarify these types of mutations using a certain DNA strand as an example and the changes of bases which may occur in it in DNA replication.
Silent mutation. If the sixth base of a certain DNA strand is cytosine, the mRNA base copied by it is guanine. The second codon of the mRNA strand is AAG, therefore. The codon AAG codes the amino acid lysine in the process of translation in protein synthesis. But if the sixth base cytosine is replaced by the base thymine due to an error occurred in DNA replication, the sixth base of the complementary mRNA strand will be adenine. The second codon of the mRNA strand becomes AAA due to this mutation instead of AAG. But the codon AAA also codes the amino acid lysine as AAG. Therefore, no change of the protein occurs though a mutation has occurred. This type of mutations are called silent mutations in molecular biology. Missense mutations. Think about a situation that sixth base cytosine of this DNA strand is replaced by the base guanine. Then the sixth base of the mRNA strand becomes cytosine and the second codon becomes AAC. The codon AAC codes the amino acid asparagine. Since asparagine is added to the amino acid sequence instead of lysine, the structure of the protein produced will also be changed. It will be a non-functional protein.
Sickle cell anemia found in human population is an example for a missense mutation.
Nonsense mutation. If the fourth base of this DNA strand, thymine, is substituted by adenine, the second codon of the mRNA strand becomes a stop codon, UAG. There are three stop codons as UUA, UAG, and UGA. When one of these codons meet, the translation process is stopped. An immature shorter polypeptide is formed due to this type of mutations. Beta thalassemia found in human population is an example for nonsense mutation.
Insertion. Addition of one or more nucleotide pairs to an existing DNA strand in DNA replication is called insertion. The reading frame of the mRNA strand copied by this mutated DNA is shifted rightward due to an insertion. Missense or nonsense mutations can occur due to insertion too. Non-functional protein is produced causing a missense mutation due to insertion. A stop codon may also be formed due to insertion causing a nonsense mutation. If the base guanine is inserted to this DNA strand as the fourth base, the second codon copied by it becomes CAA. If the base adenine is inserted to the DNA strand as the fourth base of the mRNA strand becomes uracil. A stop codon UAA is created due to this insertion, causing a nonsense mutation.
Deletion. One or two bases of an existing DNA strand may be lost in DNA replication. The reading frame is shifted leftward due to this type of deletion. Missense and nonsense mutations may occur due to deletion too.
If the fourth base of thymine of this DNA strand is deleted in DNA replication, the second codon of the mRNA strand copied by it becomes AGU, and it codes amino acid serine, causing a missense mutation. A similar situation occurs if the seventh base adenine of this DNA strand is deleted. The third codon becomes UUU, causing a missense mutation. A stop codon may also be formed due to deletion, causing a nonsense mutation. Cystic fibrosis is caused due to this type of deletion. You may understand now, silent missense and nonsense mutations occur due to gene mutations such as substitution, insertion, and deletion. Hope to discuss about chromosomal mutations through our next video. Model questions. Question number 1. Gene mutations 1. May occur due to the errors of DNA replication. 2. May occur due to strong bases or acids. 3. Are always harmful. 4. Cause genetic disorders. 5. Are transmitted from one generation to another. Question number 2. The phenomenon that cannot be taken place due to substitution. 1. Occurrence of a silent mutation. 2. Occurrence of a missense mutation. 3. Occurrence of a nonsense mutation. 4. Shifting of the reading frame. 5. Synthesis of an immature protein. Question number 3. Incorrect statement or statements about missense mutations. A. They may occur due to substitution, deletion, or insertion. B. Cause genetic disorders in humans. C. The entire sequence of amino acids of the newly made polypeptide is changed due to these mutations. D. Thalassemia found in human population is caused due to a missense mutation. E. Though they occur due to substitution, they are not formed due to deletion or insertion. Question number 4. Nonsense mutation. 1. Is a type of gene mutations. 2. Occur due to a newly made stop codon. 3. Cause a synthesis of immature protein. 4. Cause sickle cell anemia. 5. May occur due to substitution, insertion, or deletion. Question number 5. When a sequence of codons of a mRNA strand is considered as a reading frame, it can be shifted due to 1. Substitution and deletion. 2. Insertion and inversion. 3. Substitution and insertion. 4. Insertion and deletion. 5. Duplication and translocation.